Hello, in this video we are going to talk about cells. We will first start out by talking about what cells are and how we study them. Then we will move on and talk about the components of cells and their jobs and what they look like. And then we will finish up talking about how cells are connected and what those different connections do. So remember, you can pause this video at any time you would like. Make sure you are taking notes, and let's get started. All right, so studying cells. We are going to first talk about the role of cells in an organism. Then we will talk about microscopes, and that's how we study cells. And then we're going to summarize something called cell theory. So all living things are composed of cells, and no, they are not the same type of cell over and over. Just take a look at the three images below. Those are all different types of cells. In single-celled organisms, the cell is everything because that is their body. That's all that they are. That is their self. So your body, it's not a single-celled organism, right? You are a multi-celled organism, but you have different types of cells in your body. Think of it as a house. So your house is made of wood and nails. It's made of insulation, roofing, electrical wires, lighting, fans, plumbing. There are different things that make up a house, and they all have different jobs to do, just like the cells in your body. So let's think back to chapter 1 where we talked about the organization of life. So the cell is the basic unit. So one cell is considered alive. It's an organism that is the smallest organism that we can have is a single cell. So it's a basic unit. So when we get cells that are similar, so like in your body, when we get cells that are similar and they are organized together, that would be called a tissue. So a tissue are all the same types of cells together. Now, if we have several different tissues combined together, they will form an organ. And then multiple organs together form an organ system. And then an organ system will combine with other organ systems to make an organism, in our case, in our multicellular case. So if you take a look at this image down here, cell, tissue, organ, organ system, organism, that's how we are arranged, okay? So let's keep that in mind when we're going over cells, that number one, the cell is the basic unit, and then number two, that cells don't look all the same, even though we're going to see a lot of the same picture of the same cell. Just keep in mind that cells are in fact different when we look at different parts of the body. Okay, so studying cells. So we use microscopes to study cells because cells are small and we can't really see them with the naked eye. So microscopes make it much easier for us to see them. So if we break down the word microscope, micro means small and scope means to look at. So it's a small looking device, right? We can take pictures with microscopes and we call those micrographs. Now down here we're looking at two different types of microscopes, and we're going to get more, we'll get into detail here in a second, but I just want to point out that when you take a biology lab at a university or a college, you will typically use this one right here that I'm circling. This is a compound microscope, and it uses light, mirrors, and lenses to magnify and produce an image of whatever we're looking at on the slide here. This one over here is an electron microscope, which is typically used for research purposes. So two things are really important when we talk about microscopes. The first one is magnification. So magnification is the process of enlarging an object. So like zooming in, and we can make the object appear larger. Resolving power is also very important. Now I'm sure you've experienced this with your cell phone camera. If you ever tried to zoom in with your camera to something that's far away, where you're magnifying something that's far away, the image starts to look blurry. So the resolving power is not good. The resolution is low. But with microscopes, we have the ability to create a better resolution by dialing in our knobs. So we can create a nice crisp image with a higher resolution. So let's take a look here at some different pictures of magnification. So this is the same thing. This is at 5 times magnification, so that's called a scanning objective. It's typically 4 or 5 times. It is the smallest objective that you use on a microscope, and that's the one that your teacher will tell you to use when you place a slide on the microscope for a first time, just so that you can find what you're looking for and you can make it a little bit clearer. So after you make your image clear, like this one here, you can switch to another objective, and it's called the low power. So low power is sometimes 10, or it might be 40, or it might be 32 in this case. So switching to low power will actually cause you to zoom in. So we're zooming in here on this part of what is a cross-section of a plant root. And so we can tell that this is not magnified that much, but this is really magnified. We can really see the detail in each of these cells here. So next is resolution or resolving power. This image on the left has low resolution, low resolving power. We can't really see the detail, it's fuzzy. Whereas this image on the right has great resolution, has high resolution, and we can really see the detail here. It's not fuzzy at all. So high resolution is always great when we're using microscopes to look at small microscopic things. But of course, we are not going to use electron microscopes in our lab. We will use light microscopes. So we might end up with an image like this, but that's okay because we can really still see the detail. It's not that terrible, right? Okay. So let's talk about microscopes some more. So compound light microscopes bend visible light. So typically your compound microscope will have a light source, and it's going to use that light source to illuminate what's here. And then you're going to use that, or the microscope will use that light to magnify the image by bending it and reflecting it in certain ways. Light microscopes are great for looking at living things. We can make something that's called a wet mount. You can also make something that's a wet mount with a stain, and that'll make whatever you're looking at really clear. So these are cheek cells. If we do a cheek cell swab, this is what you would look at. This is a cheek cell that has not been dyed, and then this is a cheek cell that has been dyed. So really, really faint, you'd probably have a really hard time finding this cell on your slide, but when we dye it or we stain it, it's going to be really easy to find that cell and then magnify the image and really see, the detail there. So sometimes it might be green, they might be purple, just depending upon what type of stain you use. Electron microscopes are, again, really large, and they're typically used for research purposes. You probably won't use one as an undergraduate in a biology lab, but maybe as a higher level class or even in graduate school. Electron microscopes have a really high, they achieve really high magnification and resolution by using beams of electrons. So the things that we put in an electron microscope to look at, like this red blood cell, it's going to be dead. So no living things can be put into the electron microscope to be viewed. But if you do put a living thing in there, it'll be killed by the electron beams. So there are 2 types of electron microscopes. The first one is the TEM, which is the transmission electron microscope. which helps us see the detail inside of a cell. So if we were to cut a cell in half, we could really see the detail. The other type is a scanning electron microscope, an SEM, and that provides a nice exterior view of the cell. So again, electron microscopes provide amazing detail, but it kills whatever is placed inside of it. So we can't really look at the movement or behavior of the organism. All right, so cell theory is a theory that was devised by Schleiden and Schwann, and it was proposed in the 1830s. So they came up with a few details surrounding cells. So one, they said cells are the basic units of life. Two, they said all living organisms are made of cells. And then three, all cells come from pre-existing cells. So they proposed the cell theory in the 1830s. What we're looking at here is a man named Antony von Leeuwenhoek. He used a microscope. This is a microscope, not quite like the ones we were looking at, and in the 1600s, and he used this very simple microscope to get some of the first glimpses at cells. So just some cool history there. Okay, answer this question for me. When viewing a specimen through a light microscope, scientists use blank to distinguish the individual components of cells. So we're using a light microscope. We're going to use C. So special stains are going to be used to distinguish components of cells. It makes it easier to see. Okay, and that is the end of 4.1 studying cells. So hopefully now you can describe why cells are important. Hopefully you can compare and contrast light and electron microscopy, and then summarize the cell theory. All right, 4.2, we're going to talk about prokaryotic cells. We are going to name examples of prokaryotic and eukaryotic organisms. We'll compare and contrast to those cells as well. We will describe the relative sizes of cells and explain why cells are so tiny. Okay, so cells just in common have 4 components. All cells have 4 components. One is the plasma membrane. The plasma membrane separates the interior of the cell from the exterior environment. So that allows the inside of the cell to look a lot different, maybe to have different chemical composition from the exterior world. They also have cytoplasm. So cytoplasm is the cytosol, which is the jelly-like substance, and then the other components inside the cell. Cells also have DNA, so they're going to have genetic material, which helps drive protein synthesis and different processes. And then in order to carry out protein synthesis, they have to have ribosomes. So ribosomes are all of those little dots that are our protein factories. Okay, so prokaryotic cells. Can you draw and label a prokaryotic cell from memory? So I suggest pausing the video and trying to draw one first, and then we're going to see how you did on the next slide. All right, so how did you do? Did you draw this? Maybe not. Maybe you got a few of those things, and that's a great start. This is a typical prokaryotic cell. Now I say typical, not all of them look like this. So not all prokaryotic cells will look exactly like this and have all of these parts, but these are the typical parts that you will see on different prokaryotic cells. So the prokaryotic cell has DNA, and it's located inside of a nucleoid region. So that's just a region where you could find the DNA. Sometimes the DNA is attached to the interior of the plasma membrane so that it's anchored somewhere and it doesn't just float around. Prokaryotic cells also have ribosomes, so remember we have to have ribosomes. The cell membrane is another component that we have to have. So the cell membrane is this innermost wall here that you see. And then some other structures are a cell wall. So that's on the outside of the plasma membrane or the cell membrane. And then there's another layer called the capsule. So the capsule is a very sticky substance, and that helps it stick to surfaces and stick to other bacteria or other prokaryotic cells. Some prokaryotic cells have pili, which allow it to connect to other prokaryotic cells. They might also have cilia, which is not here, but that allows them to move around. They might have a flagellum, which allows them to move around. Cilia are typically small, like these pili. Flagellum are much larger. And then, let's see. I think that's all I wanted to go over. So again, not all prokaryotic cells have every single one of these components, but these are the things that you will most likely find on all of them. Prokaryotic cells are much smaller than other cells. So here's a bacteria cell, and then here is an animal and plant cell. So bacteria cells are roughly the size of a mitochondria. So if we think about the mitochondria inside these animal cells here and the plant cells here, very, very tiny, right? You can fit a lot of them inside of an animal and plant cell. So the reason bacteria are so small is because they lack a lot of the modifications that other eukaryotic cells have. And to be small, that means that they can import and export substances very easily and quickly without the need for other specialized components like organelles. Okay, so let's talk about surface area to volume ratio. So as cells get bigger, their volume increases much faster than their surface area does. So in a cell that is this size here, so 30 microns, and cells that are this size, so take up the same space, but they're only 10 microns, so if we divided it into 27 cells, divided this one cell into 27 cells, the surface area is much, much larger, 16,200 versus only 5,400. So the surface area is important to be large because the surface area is encompassed with the cell membrane. So the cell membrane, remember, it provides that barrier to the outside surface, but it is actually more important that it imports and exports substances into the cell. So if you can't import enough substances like this cell here, the cell will likely die. But these cells, being smaller, they can import substances fast enough that the cell will remain living. Plus, they probably have a lot less to maintain versus this large cell. There's a lot more here for this one cell to maintain. So it's better that cells are small so that we can keep that surface area. Large cells likely won't survive. So here's a little experiment that we've done in the past. And what you do is you take different size cubes of potato and you soak them in an iodine liquid. So this dyes the potato a black purplish color. You leave them in this iodine liquid for quite some time, and then when time is up, you take them out and you cut the potato in half to see how far the liquid made it inside of the potato. So we have our control here, which was soaked in just water. Notice that it's not purple. Our smallest cell, I put quotation marks, cell, our smallest potato is completely purple all the way through. The next largest potato has a little bit of white shading still here in the center, and then same with this one. And then our next largest potato, which is this guy here, has a lot of white still in the center, and then our largest potato has the most white. So you can see that substances aren't able to enter the cell, the potato cube, at an adequate rate, right? The smaller the cell is, or the smaller the potato is, the substance can enter fairly quickly. So smaller the better. Okay, poll question. Prokaryotes depend on what to obtain some materials and to get rid of wastes? So maybe you don't know this term yet, or maybe you've heard of it before, but it is diffusion. So diffusion is what causes substances to enter and exit the cell. And we will get more into diffusion in chapter 5. All right, so that's the end of prokaryotic cells. So hopefully now you can name some examples of prokaryotic cells. So that's like bacteria and archaeans. Eukaryotic cells like plants and animals. Hopefully you can compare and contrast especially the size of prokaryotic cells and the components. Describe the sizes of cells, and hopefully now you can describe why cells must be small. All right, moving on to eukaryotic cells. So eukaryotic cells, we're going to describe the structure of them. We will compare them, compare plants and animal cells. We'll also talk about the plasma membrane some more and summarize the functions of major cell organelles. So these are our plant and animal cells. If you want, you can take a screenshot of this page and print it. And what you can do is write a quick little description for each of these organelles or parts of the cell. So that would be a nice little cheat sheet to keep nearby for you to study. Okay. So eukaryotic cells. So if we talk about just animal cells here for a second, I just want to point out that not all animal cells are like this one here. Okay, so this is the typical animal cell that we will see in the textbook and throughout the PowerPoint, but just keep in mind that not all animal cells look like that. So there's animal cells that are fibroblasts, which is connective tissue, red blood cells, erythrocytes. There's epithelial cells like your skin, or they're in glands. There are fat cells, adipose cells, macrophages, which are white blood cells, nerve cells, sperm, reproductive gamete cells, muscle cells, right? So these are all very different from the animal cell picture that we just saw, but they are still animal cells, and they typically have the same components as that original animal cell. They just look a little different. Okay, so organelles and structures common to plant and animal cells. The things with the black bullet are found in both types of cells: plasma membrane, cytoplasm, cytosol, nucleus, chromatin, nucleolus, ribosomes, mitochondria. These are all things that are found in plant and animal cells. The things with a blue bullet, peroxisomes, vesicles, centrosome, and lysosome, are things only found in animal cells. And then the things with the green bullet, cell wall, chloroplast, central vacuole, are things that are only found in plant cells. Okay, so keep that in mind. So I just want to point out, mitochondria are found in both types of cells. Both types of cells need a mitochondria to make energy. Okay, so here's that image again, now with the definitions placed in. So hopefully you printed that original image, maybe you made some notes really quick, or maybe you're just waiting for us to move on. But anyway, this is what we're aiming for. Okay, so let's start with the plasma membrane. The plasma membrane, remember, is made up of phospholipids, which is a type of fat and oil, or a type of lipid. It has that glycerol and phosphate head with the two fatty acid tails. Remember that the phospholipid head likes water. It's hydrophilic. And then the tails are hydrophobic. They do not like water. So that creates a very dynamic cell barrier. And the cell barrier is not just made up of these phospholipids. There are also proteins that might go through the entire membrane. There might be proteins that are only on one side. There's cholesterol here in the membrane. There's things that are glycoproteins and glycolipids that are on the exterior of the cell. There might be cytoskeletal elements attaching to the plasma membrane. So it's a very dynamic structure. And we're going to get more into the plasma membrane in chapter 5, where we talk about diffusion. So just keep in mind that the plasma membrane, its major job is to control the passage of molecules, ions, water, oxygen, into and out of the cell. So we need to bring things in and keep things out, but also take things out of the cell. Okay, so the plasma membrane can be found again in animal and plant cells. All right, cytoplasm. So again, found in both types of cells. It is the region between the plasma membrane and the nuclear envelope. So in this cell, it's all the green stuff and all the yellow stuff in that cell. So it consists of a gel-like substance called cytosol, and then it's also all the things embedded in the cytosol, including the cytoskeleton. The cytoplasm is typically made up of water, and then it's made up of some chemicals as well. And those chemicals are typically like proteins and molecules, RNA, other phospholipids, DNA, and carbohydrates, polysaccharides. So those things will be embedded, but it's mostly water. And then again, the cytoplasm is again referring to the gel-like cytosol plus all the organelles in it. So next is the nucleus. There's usually just one nucleus per cell. The nucleus is found in animal and plant cells. It's typically the large circle thing that you see when you look at a cell under a microscope. It contains the DNA, the chromatin. Chromatin is DNA plus proteins. So chromatin, it also has a nucleolus in the center of it. Sometimes the nucleus is bigger than most prokaryotic cells. So remember, our prokaryotic cell was roughly the size of the mitochondria. The nucleus is even bigger than that. The nucleus has nuclear pores, which allow RNA to exit, and it is connected. The nuclear envelope is usually connected to the endoplasmic reticulum, which is here and is located around the exterior there in those two images. So the nuclear envelope has pores, and the pores look like this, and they are very selective into what comes into the nucleus and what exits the nucleus. So they're not going to allow DNA to leave the nucleus through the pores. Not going to happen. DNA stays inside the nucleus. What can leave the nucleus is RNA, and that happens through a process through these nuclear pores, which we're not going to get into. But the nuclear envelope is a double membrane, and that helps provide some protection, and it helps provide some separation for the DNA from the rest of the cell. The nucleolus is that small dark region inside the nucleus, and that's where ribosomes are assembled, and those ribosomes will leave through the pores and then go to either the endoplasmic reticulum, or the ribosomes will be somewhere in the cytosol. Okay, so more about ribosomes. Ribosomes are two units together. There's a large unit and there's a small unit. And when they're working, they create proteins. So these proteins will come out the top here as an RNA molecule moves through it. So if ribosomes were to stop working, the cell would stop producing proteins, and that's not good, right? Remember, the central dogma is DNA to RNA to protein, and that's how cells live and regulate themselves. So if our ribosomes aren't working, then the cell is likely to die. And a lot of medicines that you take, especially antibiotics, they target specific ribosomes that prokaryotes tend to have. And so if they can target these ribosomes and shut them down, then that's going to kill the prokaryotic cell that might be giving you a bacterial infection. So just something interesting to think about, that we can use the molecules that shut ribosomes down to our advantage. Mitochondria. Okay, so the powerhouse of the cell, right? So the mitochondria is found again in both types of cells, plants and animals. There's typically multiple mitochondria inside each cell. 2 is probably not enough for these cells, but these are just simplified images. They have 2 membranes. One membrane is smooth, the other membrane is very folded to create more surface area. And that surface area helps create ATP. So the major goal of the mitochondria is to create energy for the rest of the cell in return for like food supplies, right? So it has a nice relationship with the rest of the cell. Okay, poll question. So ribosomes are abundant in red blood cells. What other cells of the body would you find them in great abundance? So remember, ribosomes make protein. So where would you find a lot of ribosomes? Well, that would be in muscle cells, right? Muscles are made of protein, and so you would find a lot of ribosomes in muscle cells. Okay, so let's compare and contrast plant and animal cells really quick before we move on. Animal cells have all the things that we've covered so far, plus a centrosome, lysosome, and peroxisomes. Plant cells don't have those things. Instead, plant cells have a cell wall, chloroplasts, plastids, like a central vacuole. So animal cells don't have those things. So we're going to get into those special components that each of those have, and I will try to point out which one has which. Okay, so peroxisomes are actually found, there's some new research out that peroxisomes are in plant cells, not in quite as high abundance as animal cells, but mostly found in animal cells, okay? Peroxisomes are small, round organelles. They have a single membrane, and their major job is to break down fatty acids, amino acids, and poisons. So one poison that you might consume is alcohol. So yes, alcohol is a poison for your body. Your liver tends to detoxify it. So your liver, your organ, your liver, tends to have a lot of peroxisomes inside the cells that make up the liver. So they will break down fatty acids, amino acids, and poisons like alcohol. They're called peroxisomes because they create H2O2, which is the chemical formula for hydrogen peroxide. And then that H2O2 is further broken down into oxygen and water. Now, centrosomes are only found in animal cells. They are located right here. The centrosome consists of 2 centrioles, and they lie at right angles to each other. Each centrosome, or each centriole, has tubulin protein, which are part of the cytoskeleton, and their major job is to pull chromosomes apart during cell division. All right, plant cell walls. So plant cell walls only found in plants. They are the tough exterior portion of the plant. It makes plants hard to digest. but it also provides support for the plant. So plants don't have bones, but they can produce these tough cell walls to help protect and support them. So plant cell walls are made up of cellulose, which is a very large polysaccharide, and you can really make out the cell walls here in this image of a onion root. Cell walls don't do anything in terms of letting substances in and out. The plant cell wall's only job is really to provide structure and protection. Okay, chloroplasts. So chloroplasts are those green blobs that we'll learn about later on in the semester. They have their own ribosomes and DNA, so they were thought to be bacteria before, just like mitochondria thought to be a bacteria before eukaryotic cells came around. They are made up of 3 membranes. They have an outer membrane, inner membrane, and then there's membranes for these thylakoids. The thylakoids is where some of the reactions of photosynthesis happen. The other reactions of photosynthesis just happen in the liquid that makes up the chloroplasts. Typically, chloroplasts' major goal is to create sugar. So they're going to create sugar. They also create oxygen, which is released to the air. But once they create sugar, the sugar is sent over to the mitochondria, which can break it down for energy, for ATP. Plant cells also have a central vacuole, which usually houses water, and that water can provide some turgor pressure for the plant cell itself. So if that vacuole dries up, maybe you haven't watered your plant for a while, it starts to wilt. What's actually happening on the cellular level is that the cell membrane is pulling away from the cell wall. So once you water your plant, it will cause that vacuole to expand, and that pushes the cell membrane closer to the cell wall. And then if you water it too much, it might make the cell wall kind of bulge out a little bit. Mostly central vacuoles house water, but they might also house some dissolved minerals as well. Okay, poll question. So which of the following is not found in animal and plant cells? So that would be the vacuole, right? So that's not found in both, it's only found in plant cells. Good job. All right, so that's the end of the eukaryotic cell section. We are going to get into another part of the eukaryotic cell. You probably figured out that we haven't covered everything yet. So we're going to get into the rest here in the next section. Okay, so 4.4 is the endomembrane system in proteins. So this system functions to create protein for use for the cell itself or for export. So we're going to talk about the components of the endomembrane system, and then we will talk about the relationship of each part. Okay, so the endomembrane system is what we're looking at here on the right. It consists of the rough ER, the Golgi apparatus, vesicles, and then the plasma membrane. So the endomembrane system works together to create, modify, package, and transport lipids and proteins. Now, one portion that's not listed here is the smooth endoplasmic reticulum, not in the picture, but we will talk about it here in a second. All right, so first we're going to talk about the endoplasmic reticulum. There are 2 types. One is the rough ER and one is the smooth ER. The rough ER has ribosomes embedded on the surface, which makes it rough in appearance, and it makes proteins because it has ribosomes. The smooth ER does not have ribosomes, and so it's going to make lipids instead. So the rough ER, the smooth ER, either one, or both of them in fact, have a hollow portion, and that's where the substance, the protein or the lipid, goes when it's created. And once it's created, it breaks off and goes to where it needs to go. The endoplasmic reticulum is typically continuous with the nuclear envelope. So the first type that we'll talk about is the rough ER. The rough ER, again, is rough because it has ribosomes on it, and those ribosomes create protein. And so those proteins will enter the hollow lumen of the RER, and they will exit via vesicle and enter the Golgi apparatus from there. So again, remember, proteins can be hormones, they can be enzymes, they can be structural proteins, they can do a lot of things. They can be embedded on the membrane here at the end as well. So the RER can also make portions of phospholipids, and the phospholipid portion is added to the other portion, which is created by the smooth endoplasmic reticulum. So first, let me point out how smooth the endoplasmic reticulum looks. It does not have ribosomes. So instead of making protein, it synthesizes some other things like the other portion of phospholipids. It also makes steroids, which is, remember, a lipid. It can synthesize carbohydrates as well. The smooth ER can help detoxify some medications and poisons. One thing, like if you take allergy medicine on a daily basis like myself, your cells probably have some really strong smooth ER organelles in them because that helps detoxify medications. It also stores calcium ions in muscle cells, and calcium helps muscles move and contract. Okay, so next is the Golgi apparatus. So after the RER or the SER creates lipid or protein, it's going to be sent off to the Golgi apparatus. So the Golgi, I like to call it like a stack of pancakes because that's kind of what it looks like. And it has these empty spaces as well. So the Golgi helps to sort and package and tag the Golgi apparatus. I like to think of it as like UPS or the Postal Service or whoever. They help, the Golgi helps determine where that lipid or protein needs to go. So it's like a sorting facility, right? A packing and sorting facility. If the Golgi finds an error in the lipid or the protein, it's going to send it off to a lysosome to be broken down into its components, and then those components can be reused by the SER and the RER to make a protein or a lipid again. So lysosomes, what are they? So they're little vesicles that contain some enzymes, and they help break down molecules or worn out organelles. Typically, they break down molecules that are not created correctly, or they might be poison, or they just don't work correctly anymore. Okay, so can we trace the path that a protein would follow? So first I want to point out that ribosomes are not only found on the endomembrane system. Ribosomes are also out in the cytoplasm. If a ribosome in the cytoplasm creates a protein, that protein will typically stay in the cytoplasm. However, if a ribosome on the RER creates a protein, then it's subjected to the endomembrane system. So once that protein is created, it's going to be sent off via a vesicle to the Golgi apparatus, which it will travel through and be sorted and tagged for somewhere to go. And if it's not broken, right, if it's broken, it's going to be sent to a lysosome. If it's not broken, it'll be exported from the Golgi apparatus and be sent to where it needs to go via a vesicle. So this one is a protein that needs to stay on the surface of the cell membrane. Okay, so poll question. What happens to proteins synthesized by ribosomes on the RER? They're going to be what? D, right? Transported to the Golgi. All right, another poll question. A cell that has a higher average of Golgi bodies within its cell, what would be the advantage? That would be C, to secrete more material. So the more packing and sorting we can do, the more we can send materials out. So maybe this cell is a gland cell, like a hormonal gland cell, and it's creating materials, and it needs to secrete a lot of materials all the time. Okay, so here's a nice sum up page, and it helps show some pictures, and it gives you some short little definitions or analogies of what the organelle does for the cell. The things that are boxed in red are for animal cells. The things that are in green are in plant cells. And then the things in blue are in all eukaryotic cells. There is a quizzes that I created that goes over all the cell organelles and what they do for the cell. So if you would like to play that, there's the link. And that's the end of the endomembrane system. So hopefully you can list the components, which are the nuclear envelope, the RER, the SER, the Golgi, vesicles, lysosomes, and the plasma membrane. And now hopefully you can recognize the relationship that each of those components have to each other. All right, so next we're going to talk about the cytoskeleton. We will first describe what it is and describe what makes up the cytoskeleton, and then we'll talk about what uses it has. So the cytoskeleton is a network of protein fibers. It helps maintain the shape of the cell. It gives it a rigid structure, but it also helps hold organelles in place, and it allows movement within the cytoplasm, and it enables cells to have movement within like a multicellular organism, or if they're single-celled, it might be like part of the flagella or the cilia, and it allows them to move. So this is a nice picture here of the cytoskeleton. It has microfilaments, intermediate filaments, and microtubules. So those are the three components of the cytoskeleton. And here's another picture that summarizes where you could actually find them inside the cell. So intermediate filaments are like an intermediate size compared to the other two types. You can find them throughout the whole cell. The microfilaments, which are very small, you can find them on the exterior portion, and then microtubules are in the interior, and they're typically only created during cell division. They help pull the chromosomes to either pole of the cell. So intermediate filaments provide anchors for organelles, and microfilaments provide support for the plasma membrane. So here's what microfilaments look like. So these are the very small ones on the exterior portion. They only consist of 2 strands of protein substance. They are called actin. You don't need to know that, but they help provide some support and help stabilize the round shape of an animal cell. Microtubules, again, are mostly just for movement during cell division. They are very large. They form a tube, a hollow tube, and they are made up of tubulin, so that's why they're called microtubules. Intermediate filaments are solid. They look like those Twizzler pull-apart candies, and they help anchor the cell organelles, and you can find them throughout the entire cell. So cilia and flagella are made up of microtubules. So remember, they're those hollow tubes. They are very large, and when they are attached to like motor proteins, they can move. And so cilia are made up of shorter microtubules, and they just kind of whip or a beating motion, which allows movement. And then flagellum are typically beat in a propeller-like motion, and that allows movement as well. So that's it for the cytoskeleton, pretty short and sweet. So hopefully now you can describe what the cytoskeleton is and its job, and then hopefully you can compare and contrast those three types of filaments, and hopefully you can compare and contrast cilia and flagella. Okay, last but not least are cell connections or cell junctions. We're going to talk about the extracellular matrix. We will talk about how plant and animal cells are connected and how they communicate. So the extracellular matrix is on the exterior portion of the cell. So in a plant or an animal, cells are not usually butted up to each other end to end. There's usually some space between cells, and that allows for like fluids and things, nutrients, waste products to move around. Right, so we want waste and nutrients to come by the cell. So in plant cells, the extracellular matrix is usually made up of the cell wall, and that helps provide support. The cell wall is not usually completely solid, so there still is some movement for wastes and nutrients. In animals, there's usually some like collagen protein, other types of linking proteins, or connecting proteins on the exterior portion of the cell. So that helps create a nice a nice tissue, so a cohesive tissue. It keeps the tissue together, so it anchors one cell to another cell. But yeah, so that's the extracellular matrix. So cells are connected to one another. They are connected in various numbers of ways if you're an animal. In plant cells, they're really only connected in one way, through plasmodesmata. And in plasmodesmata, there is a gap in the cell wall. And the gap in the cell wall allows the cell membrane of adjacent cells to butt up to one another so that they can communicate. And what I mean by communicate is allow materials to move from one cell to another. So here's a lot of plant cells here. There's 1, there's 2, there's 3, there's 4, there's 5. So there's this big gaping hole in the cell wall, and that allows for materials to move through. So plant cells, the only channel, really, the only connection they have is plasmodesmata. Now, animal cells have 3 types. There's a tight junction, a desmosome, and a gap junction. So the first one is a tight junction. A tight junction provides a watertight seal between adjacent animal cells, and that helps prevent materials from leaking. So in cells like your skin, you would not want the liquid inside your body to leak out onto the exterior portion of your skin. So the tight junction provides that barrier for your skin. So all the skin cells are connected through a tight junction. Desmosomes are also called spot welds. You could think of them as like a button, sort of, where they're tightly together, but there's some gaps here between desmosomes. So this allows for stretching, and they're found mostly like in the heart, lungs, and muscles where you would want some stretching to occur. So tight junctions would not be the best for this because they won't really stretch that much, whereas desmosomes can stretch a little bit, but they still anchor the cell to another cell. The last one, the last junction found in animal cells is a gap junction. This one is like a plasmodesmata. It provides some communication between adjacent cells. So there's a little channel here so that material in one cell can exit and enter another cell. So there's a nice summary page of the different types of junctions in animals and plant cells. And that's the end of 4.6. So hopefully now you can describe what extracellular matrix means and list different ways that plants and animal cells communicate. And that is it for chapter 4. So hopefully you learned about cells, how we can study cells, what cells are, the difference between prokaryotic, eukaryotic cells, and then the difference between plants and animal cells, which are eukaryotes. If not, go back through the video. There are some videos and links that I put throughout my PowerPoint that you can enter into your search and find them. There's Quizlets and there are interactives, so feel free to play them. But I hope you have a great day.